Day, everyone. My name is Jerilyn Calaor. I was born and raised on Guam, graduated from UOG's biology program in 2018, and now I'm a PhD student at Iowa State University. Today, I'll be talking a little bit about my dissertation research, which explores the role of birds in shaping arthropod food web dynamics. First, I'd like to recognize that this work is part of a larger project across the Marianas, Hawaii, and Okinawa. So I'd like to acknowledge the suite of people involved in the Pacific Bioscanner collaboration, and they're highlighted here. So diving in, predation plays an important role in shaping communities. Here we have a simplified terrestrial food web with a bird as a top predator, and top predators often serve the role of maintaining prey populations, such as arthropods in this case. Arthropods, in turn, have important interactions with plants, such as herbivory and pollination. And so birds can indirectly affect the plant community through their direct effects on the arthropod community. And so, given this, a change in bird communities, let's say by the introduction of the brown tree snake, as we have seen on Guam, can likely have impacts that cascade through the food web. And so, this begs the question, how does bird loss alter arthropod food web dynamics? And people have begun to explore this question through the use of bird exclusion experiments, which involves using bird netting around part of the plants while leaving other parts open to bird access, and then comparing responses of arthropods and plants in the presence and absence of birds. And the current state of literature have explored these parts of the food web here. And some things we know are that spiders increase without birds. Plants generally benefit from birds and experience less herbivory and increased plant biomass, and the effects vary with season, geography, and land management. What remains relatively unexplored here is this key gap of our understanding of the food web, and it's this interaction between predatory and herbivorous arthropods. And it's also important to test if these patterns from these smaller scale studies using these reviews and meta-analyses scale up to a landscape level. And this is where the unique system of the Marianas comes in. And so just diving into some things we already know about the dynamics in our system, is that birds seem to have inconsistent roles on seedling survival. And so here we have data on survival in the presence and absence of birds. Our x-axis has plant species and our y has the proportion of seedling survival. Blue lines are responses in the presence of birds and green lines in the absence. And this is showing trends on Guam versus islands with birds, but the trends are also similar to an exposure experiment done only on islands with birds. And so to dissect this a little bit, what we're seeing is that Aglaya has seedling survival higher in the presence of birds. But the opposite is true for Marinda, where seedling survival is higher when birds are not present. And the same is the case for Nisosperma. And so what we can kind of take home from these trends here is that there's this inconsistent role of birds on survival, and the responses seem to be more species specific. So it seems to be a little bit more complicated. But what is consistent is spiders. And specifically, we're seeing that spider webs on Guam are more abundant and they lack seasonality. And so again, this just reinforces this need to revisit this gap here in our food web. And so to do that, we ask two questions. First, we ask, how does bird loss impact arthropod community composition? And second, how does the diet of predatory arthropods change in a defaunated system relative to a faunally intact system? And to begin to answer these, we sampled arthropods along set transects in Saipan, Tinian, Rota, and Guam in the summer of 2019. And at each sampling point, we collected samples from both the vegetation and ground. Specifically for vegetation sampling, we used beating sheets where we beaded vegetation and collected arthropods that fell in a beating sheet using an aspirator. And for ground sampling, we laid out meter squared quadrats, collected all of the litter that fell within the quadrat, immediately put it in a leaf sifter, and then further refined them in a Winkler funnel for 2 days. And arthropods were collected in 95% ethanol for both of these sampling methods. So now we have a bunch of samples. And for the community assemblage question, we're leaving these samples in bulk. But for the second question regarding diet, we're sorting predators such as spiders, lady beetles, and ants from our bulk samples. And once they're sorted, we're isolating the abdomen from these predators to, again, hone in on their gut content. And so these samples, whether bulk or sorted, are then subjected to this meta barcoding workflow here to prepare DNA libraries for Illumina sequencing. And we're specifically targeting the CO1 region. Um, and metabarcoding is advantageous because comparing arthropods using traditional methods can be extremely time consuming and requires some expertise in insect identification to get better taxonomic resolution. Metabarcoding, on the other hand, allows for quicker identification, and there's utility in employing undergrads even if they have little experience working with arthropods. And so, with that said, I would like to thank the great team of undergrad researchers who have been part of this work. And so, these suite of people have definitely helped so much in processing the samples and moving this meta barcoding workflow along. Um, and so Illumina sequencing will yield sequences in a FASTQ format, and then we follow this bioinformatics pipeline to create what we call an OTU table. And this involves blasting our sequences to databases linked to NCBI, which includes databases such as GenBank that has species from the Marianas. And so we get something like this, where we have sample information that includes some information about site or collection method. We get a similarity score, which tells us how similar our sequences are to the sequences that they match with in the database. And then we get a taxonomic identification. And then we have the number of hits for each of these OTUs in each sample. And I've been speaking about OTUs. So first, let's backtrack. And an OTU stands for Operational Taxonomic Unit, and it's used to classify groups of closely related individuals. And they are equivalent to, but not necessarily in line with, classical taxonomy. And so that's why we're referring to them as OTUs. Um, and these OTU tables can then be analyzed to look at differences in community assemblage and predator-prey networks. And so far, we have sequenced samples collected from Rhoda. So all of the preliminary results I'll be presenting today are of Rhoda alone. And so let's dive into some of those results. And so preliminarily, we're finding that beating sheets and litter samples cluster distinctly when using these ordination methods. To dissect that a little bit, each point here represents a sampling point along our transect, and they are colored by site, and we had 6 sites in Rhoda. And the shapes of the points represent the collection method, where we have circles representing our beating sheet samples, and triangles representing our Winkler funnel samples. And what we're seeing here is that they form these distinct clusters by shape. So we're seeing that the arthropod community found in the trees is distinct from that found in the soil, which is kind of cool and implies that we're getting a more comprehensive look at the community given our 2 sampling methods. And then we can map these data onto a network. And we have our taxa here on the top, and they're identified to order. We do have better taxonomic resolution for our OTUs, but leaving it to order kind of helps us see this story a little bit clearer. And the bottom group here represent our sites. And again, we had 6 sites in Rhoda. And each line here that connects the taxa to the site represents the presence of that taxa at that site. And the width of the line illustrates an abundance of that taxa. And so what we're seeing here is that the abundant taxa in our communities on Rhoda seem to be beetles, Hymenoptera, which are predominantly ant, and Socooptera, which are the book and bark lice. And to this network, we can then add the diets of spiders. So these are the occurrences of the taxa found in the abdomen of our spiders. And these spiders were collected at the same points as the bulk community. And so what we kind of see here is that Hymenoptera are also common in the spider diets. And you can kind of see this as the lines connecting the Hymenoptera to the gut content samples on top are relatively as wide as the ones on the bottom. So it seems to be that Hymenoptera are both represented abundantly in the gut content as well as in the bulk community. Um, Contrastingly, beetles and lice seem to be represented in the diet, but they're not as common in diets as they are in the bulk community. And you can kind of see that because the lines connecting them on the top seem to be thinner than the ones that connect them to the sides. Um, Rather, Lepidoptera, Diptera, and Collembola seem to be making this larger part of the spider diet, although they are not as abundant in the bulk community. And this is really neat because it kind of shows some selectivity of prey happening with these spiders. Um, and it's important to note that we aren't getting occurrences of spiders in our gut content samples because we have used primers that specifically block our spiders. And yeah, so this is kind of what's happening in Rhoda so far. And to get at the questions posed in the beginning, we're currently working on prepping our libraries of samples collected from Saipan, Tinian, and Guam. And so far, we can only report the numbers of predators pulled from each of these samples. And so here we're seeing that Guam has the highest number of spiders pulled from our samples, which is neat because the paper in 2012 only looked at spider webs, and most of the spiders that we're pulling from our samples here are actually non-web builders. And so this is kind of showing that the non-web builders may be following the same trends that we've seen with the webs, which I think is a pretty neat story there. And so once we have our library sequence, we can then make inferences about the role of birds by comparing Guam to the islands with birds. Um, and so, yeah, we hope to eventually get answers as to what's happening in the arthropod community and interactions that change within this black box here to then help us understand how bird loss might affect the overall forest community. And so, again, I just like to thank everybody involved in this project in one form of or another. And I'd be happy to take any questions. Suzy Maasi.